All right. Okay, I think that's working now. So as Amy mentioned, I'm a clinical geneticist, so I'm a doctor. I'm also a researcher. I've been working in the field of clinical genetics for over 20 years now, so for quite some time. And I'm currently based in London in the UK. So I'm going to give a little bit of the history and background. I've put in a little bit just of the genetics because I'm not sure how much information people have been given through their clinicians. Talk a little bit about what we know about the genes and the clinical features, and then how we can move forward together. So this all started several years ago when I was at the Sanger Institute, and that's just outside of Cambridge here in the UK. And there was a very large study called the Deciphering Developmental Disorders Study that was looking at children across England and Northern Ireland to try to identify genes associated with different developmental disorders. And in that study, BCL11A came up as a candidate gene. And it was a gene that I was interested in the work that I was doing with Dias-Logan during my clinical PhD there. And it's a gene that we know is what we call a transcription factor, which actually turns on and off other genes. So it's a gene that impacts other genes. And so to try to identify whether this was really something that was causing the developmental problems, we had two approaches. So we reached out through international collaborators to identify other patients at the time. And we also had a mouse model of BCL11A at the Sanger Institute, which we characterized. So I'm not going to go through all of that work that we've done, that's published work, but just to say that we were able to show through the features that we could identify in common in the patients, but also what we found in the mouse, that it confirmed that BCL11A was having an important role in development, and that it was causing the developmental problems of these children. So we published that in this article, and that's me and Darren on the day of my PhD viva. We published this in a peer-reviewed journal. Just to say that the reason it came up as Dias-Logan syndrome is because the organization that kind of gives names to syndromes, OMIM, called it Dias-Logan syndrome. And I can just, as part of the history, say that Darren and I contacted OMIM just to convey that this was many people contributed towards this, even though it was driven by us. It was a lot of work, not by dozens, but potentially hundreds of people that brought this together. So we didn't think that it warranted, having our names. And so they gave it another disorder, which is a mouthful, called intellectual developmental disorder with persistence of fetal hemoglobin. Someone asked on the Facebook group, where does intellectual developmental disorder come from? That is the term that the World Health Organization uses to define what others will call intellectual disability or severe learning disability. So that's where the IDD comes from. Currently, we're using BCL11A developmental disorder because we feel that it's not just intellectual; it's multisystemic. There are other things in the body that are affected. But at the end of the day, I think the patients and families call it what you want, and that's fine. That's fine by me. So again, just a genetics overview. So every cell in our body has 46 chromosomes. I like to think of it like an instruction book that's so big that's an encyclopedia with 46 volumes. So we have 23 pairs of chromosomes. So most of our genes, with this exception, are in pairs. We have everything in duplicate, one from mom and one from dad. So the BCL11A gene, which is on chromosome 2, we have one copy from mom and one from dad. The only pairs that are different are the final two, the pair number 23, for people who develop as females, it's an X from mom and an X from dad. For people who develop as males, it's an X from mom and a Y from dad. And so when sperm and egg come together, each parent passes on one copy of each of those pairs. So BCL11A developmental disorder is an autosomal dominant condition. What does that mean? It means that all we need is one copy of the gene to have a variant to have the condition. So a person that's affected would have an unaffected copy and an affected copy. And that's different from conditions like cystic fibrosis, which many of you probably have heard of. That's a condition where probably many of us here are actually carriers for cystic fibrosis, where we would have one of the copies with the mutation. But for those conditions, which we call recessive, we need both of the copies with the mutation. For this type, for the dominant condition, it's like the name says, that we do need the two. There's a dominance of the mutation over the normal copy. And so if a person has an autosomal dominant condition, that means that they have one copy of the gene with the change and one without, which means they're either going to pass the copy with or without, and their partner will pass a normal copy of the gene. So for a person who is affected, and we do have people with BCL11A developmental disorder who have children, their risk is 50-50 of having at each pregnancy. So in each pregnancy, one of these 4 chances can happen, and 2 of those chances are having an affected child. Usually it's a sporadic condition. It happens for the first time in that child. And this happens by a mechanism which we call a sporadic de novo mutation, and that is thought to usually happen either within the sperm or the egg. So parents would not have that mutation, but it would be in the sperm or the egg, and then it's passed on to the zygote, and so that baby goes on to develop the condition. And what I really want to stress, especially for parents here, is that if you have a child with a de novo mutation in BCL11A, they're not different than the rest of us, and that we all have de novo changes in our genome. The main difference is that we have 3 billion letters within our genome, and the changes that we have that were not in our parents, by and large, are going to land in areas of the genome that don't have consequence. Only around 2% of the genome is coding for proteins. So they're no different in that sense. It's just sporadically, by chance, it has fallen within a gene that is important for development, and that's why they're developing the condition. And it's also important that there's nothing that we can do to cause or prevent this. So there are no known environmental factors that would lead this to increase in cause. The only known slightly increased chance for this type of de novo mutation is paternal age. So many of you will have heard of Down syndrome, which is an extra chromosome. That risk increases with maternal age. This type of de novo mutation increases slightly with paternal age, but it can happen from either parent. And at the end of the day, it doesn't matter whether it comes from mom or from dad. That has no importance to the child, actually. More rarely, there's no mutation within the first cell of the baby, but in very, very early divisions of the embryo, a mutation can occur. And then if a mutation occurs in one cell, all the cells that are dividing and deriving from that cell are going to have the same mutation. So that's what we call mosaicism. If you think about mosaics that are, tiles with lots of different colors. Mosaic in genetics means that we have cells with different genetic constitutions. So that's also a possibility of it happening. Just to note that for a de novo condition, if it's post-zygotic mosaicism, this is quite rare. The risk for the parents it's not increased. Just going back to this chance, there's a small chance that we believe is around 2% of having other sperm or egg with the same change, and there would be a 2% chance of it happening again in a second pregnancy. So what do we know about BCL11A developmental disorder? This is from a publication that I think many of you will have seen. Certainly all the patients that have gotten in touch with me, I've sent this paper. And this is just a graphic that the bigger the bar, the more frequent that feature is. So we had information from 42 patients, many of whom are your children, I believe. And the age distribution was biased towards children. So this is the number of patients, and these are the age ranges. So we only really had 2 adults over 21 years when we started that study. So with regards to the question whether we have insight into adulthood, We don't have many adults on whom we have accurate information, on whom we have clinical records, but we can talk about that if there are further questions. But I'll go through some of these features a little bit more. So there was a question about development and about speech. So the most frequent age for first words was 2 years. There were only 8 patients who were nonverbal at the time of their last assessment, but note that many of them were actually young as well. So there are some children who even in their teens are nonverbal, but most people with BCL11A developmental disorder, at least that I'm aware of, and I'm aware of plenty, do have speech. The range is really quite variable, and that's what we see here. So this is the severity, so moderate, mild, or severe. So severe intellectual developmental disorder is rare. There are many that are mild. Many are going to be moderate. So that's going to require support in school. It's going to require an educational healthcare plan. Some of these children will go to a special needs school, or at least have special support if they're in a mainstream school. We didn't know the exact severity of developmental disorders in some of them. Behavioral problems are actually quite common. So we know that even though autism is just around 30 to 40%, other behavioral problems are common, and sometimes they increase with age. I'm going to run through this one. These are just some of the facial features that we saw, but actually we don't recognize that there's a typical facial features of BCL11A developmental disorder. So we don't call it a recognizable condition like some of the other conditions that if you see them in the street, you would recognize, like Down syndrome, for example, that most people can recognize. In terms of the neurologic features, so hypotonia, that's kind of low core muscle tone. That is the most frequent one that we saw. Seizures were less frequent than we originally thought. So only a quarter of patients, so 1 in 4 seem to have seizures. And they seem to respond well to regular medication. So they're not under any special treatment plan usually for seizures. Other things that are of concern are the joint hypermobility, and that may be associated with the hypotonia as well, but usually kids will require some physiotherapy if they have the joint hypermobility. We have the increased fetal hemoglobin. This is because of the function of BCL11A in the blood. And just to note that we have no evidence that increased fetal hemoglobin has any consequence. It can actually be beneficial in some situations, and there's a condition called hereditary persistence of fetal hemoglobin caused by changes in a different gene that actually helps people who live in high altitude or if they have sickle cell disease, but because it helps oxygen capture. So we have no evidence that this is a problem, but it is a marker. And when we have a genetic change we're not sure about, measuring fetal hemoglobin helps us understand whether that genetic change is causing an issue with the gene. Scoliosis is another that I'll talk about later in terms of the management. Unfortunately, congenital malformations are quite rare. Just a little bit about the type of genetic variation. So DNA is our genetic code. That codes for this intermediate molecule called RNA, and that makes the protein. So the first type of variant that we found were missense variant, and those are changes that change the spelling of the instruction, but it still makes a protein, but it's an altered protein. And in BCL11A, we think it's sufficiently altered that it just doesn't do its job properly. The most common type of change is actually a loss of function variant, and that one is going to cause a stop in the instruction. So this instruction is being read to make the protein. It will at some point stop and just not make the protein, or make much less of the protein. So it's what we call a condition that's haploinsufficient. Haplo with half, insufficient, it's not sufficient. So only one copy of the gene, only half of the amount that people usually have, is not enough for normal development. There were questions as well about what do we understand about the specific types of mutation and whether we can predict the trajectory. Not yet, but we're hoping that we may be able in the future as we have a lot more data. So genes are quite funny in that they actually don't make just one protein. So BCL11A in particular is a gene that can make lots of different sizes of the same protein. If you think about it as a very complicated curry recipe with lots of different ingredients, if you're going to be taking out or adding a few ingredients, so taking out a few of the, reading a few of the pieces or not, you're still going to get a curry, but it's going to have a slightly different flavor. So that's the way I try to explain it. It can code different sizes of the protein, and we believe that each one of different sizes and with slight different changes within them plays slightly different roles in development and in different parts of the body and in different cell types. So we're trying to understand whether the mutations, we know that some mutations are not going to affect this small isoform, for example. The isoform is the name we give to different forms of protein, but it's still early days in that understanding. So we have some preliminary evidence from the patient cohort that we saw. So looking at, this is the type of mutation. So these two here on the left, these PTV, are the ones that are loss of function. These are splice, which seem to work as loss of function. These are the missense, so that slightly changed instruction. And these are copy number variants. So these are where the gene is actually missing entirely. This is in terms of developmental delay. So we have four patients who had no intellectual disability, and then we have the others had mild, moderate, severe. It doesn't seem to change depending on the type of mutation, but again, it's still early days. When we looked at the head size, it looked like the ones who had a specific type of loss of function mutation that affected just the long isoforms, it seemed to be a little bit more severe. So this is the size of the head circumference in comparison to normal range. So just slightly smaller. Just to say that we also found a little bit of evidence with regards to the tone that was slightly different in one type of mutation, and behavioral problems as well. But again, we really do need much more data to be able to be sure about this. So with regards to management, and because this is an international audience, you know, I really need to make it clear that the management really does depend on where you live and what's accessible and what's available to you. And that's going to vary drastically depending on where you are. Even within England, it's going to vary between different councils, whether you're in the north or in the south. So what we recommend is that at diagnosis, if a patient hasn't had this already, some of them have already had this, that they have a developmental assessment to see what their needs are, to see what their challenges, but also what their strengths are. So, you know, we can build on their strengths, especially in school and in learning, to have an individualized educational plan to build on those strengths. A detailed physical examination, including a neurologic examination, also see if they have hypermobility, to check whether they have a cleft palate, an ophthalmology assessment, because 2/3 have strabismus, which is a lazy eye, which is usually not severe. Some kids have required some patching, but otherwise it hasn't been particularly severe. A hearing assessment, a behavioral assessment, and genetic counseling, so parents can understand, you know, how this happens. A little bit of what I talked about in the beginning, but in an individualized way for that family. We have no evidence of for treating patients with BCL11A developmental disorder differently than other neurodevelopmental conditions. But again, as we learn more about these rare conditions, we're hoping that we will have a little bit more of an individualized plan specific for each type of condition. So what's important is monitoring for scoliosis, because we know that up to 1/3 can have scoliosis, and it doesn't seem to be very severe, but it can cause back pain, and we want to avoid back pain. To monitor growth in feeding. Some kids have had some issues with feeding, particularly in early infancy, so to make sure they're getting the right type of nutritional support to grow well. Even though the risk of seizure seems to be low, we do recommend that there's a low threshold for investigation. And what do I mean by that? If parents or teachers or the nursery, if you suspect that there may be seizures, it's important that the pediatrician or the GP knows that and initiates an investigation. So it's taking parents' concerns seriously, because there is a risk. It's more likely that they don't have seizures. 3/4 don't, 3 and 4 don't. But if it is that they do have, you want to get that checked so you can initiate medication if possible. Educational support should be an individualized plan, again, depending on each person's strengths and difficulties. And the physiotherapy and occupational therapy is going to really depend on the degree of hypermobility and the motor delay. This is just a very short summary. I've pointed some people to the gene reviews where we have a little bit more detail, and that has some more specific kind of US considerations in terms of support. It's not very updated, I can say, but at least that part of support I think is relatively updated. So ongoing research. So within my lab and over the last few years, I've been dedicated to the clinical characterization. That's why we got all of these clinicians and researchers across the world together to get this paper together. We do have a database that is part of a larger database. That's, let me put that here. So that's the Human Disease Gene website. So that's by the University of Nijmegen in the Netherlands. It has information about not the specific mutation visible to the public, but it does have frequency of features. All of everything that's put in is curated by geneticists, which is either myself or Dr. Angela Perrone at the moment. And we did get some help from the local team at Nijmegen to put in a bit more information as well. It has the advantage that it has standardized terms across the platform, so it can be compared to other neurodevelopmental conditions. It complies with the European regulations. But it's all clinician submitted. It's a useful database, but it is not a patient registry. This is not a patient registry, it's a database. And currently, unfortunately, the platform does not offer the option for patient uploaded data. So this is not, so if you were to send me data, I would need to have your clinician send me the data for us to put it in here, actually. So let me. So in my lab, we're also looking at the interaction between BCL11A and other proteins in human cells, and we want to study the function of the different isoforms a little bit more deeply. We're not doing this yet. We're applying for funding to do this. A colleague in Germany, Stefan Britsch, he's done quite a bit of work on BCL11A in the cerebellum, which is the back part of the brain that gives us our balance, and the cortex is the part that kind of helps us think within our brain, and he uses mouse models to do that. So I did mouse models when I was working with Darren at the Sanger. Now my work is almost all in human cells. Outside of kind of these two labs that are a bit more dedicated to BCL11A, there are quite a lot of other people that have provided lots of insights into the biological function of BCL11A, and I just pointed out kind of the three main labs that have provided insights. So their research is broader. It's about brain development overall, so they don't just study BCL11A, but they've really, within their research, they've provided lots of very, very important information for us to understand how it works. So moving forward, what I hope that we can do together as time goes on is to improve the knowledge for long-term treatment. So for this, we need structured patient organizations. We need patients to organize, right? We need patients to get together and to have voices within those organizations that represent multiple different voices. So there's continuity. So, you know, if some parents for different life reasons cannot continue to work as actively in the organization, there's someone else available. And I was telling Amy about how we have some patient organizations that have been going for 30 to 40 years. And the reason for that is because they have a strong multidisciplinary board with several parents and patients as well. It's important to note that many patients with BCL11A developmental disorders will have a lot to contribute to these organizations, either as patient ambassadors or, depending on their ability to read and write and to work with IT, to work within the organization itself. And those are very important contributions to take into account. What I am hoping that we can build over the next, my aim is 3 to 5 years, is to co-design. And what I mean by co-design, it's an active engagement with clinicians, patients, and researchers, so all together, a formal patient registry that will serve everyone, and a natural history study, because they're not the same thing. A natural history study is something that is following people over the years. The patient registry is knowing who are the patients, where are they happy to be contacted, if there are studies that they can participate in. So they're slightly different things. They have slightly different ethics arrangements, and they also require different funding. For example, a natural history study is much more difficult to have a very long-term, having parents contribute data long-term. We're also hoping to advance the understanding of the biological roles. And again, my lab is primarily, I do lots of clinical work, but I do cell work as well in another gene as well, to advance the basic and translational research in BCL11A, because if you don't have the starter, you can't make a sourdough. So understanding basic biology is what's going to help us understand the condition as well. And I think that's all for me. Sorry if I took too long, Amy.
Oh, no, that was incredibly informative. I truly appreciate that. Thank you so much for being here with us today. Now, before I introduce our next speaker, if you are using those closed captions for a language other than English or French, please take a moment to go back into the settings menu and change the language of the meeting to French. This will hopefully continue accurate translation into your desired language, as our next guest will be presenting in French. And now I am pleased to introduce Virginie and Alexis, the founders of Together for Dias-Logan Syndrome. As parents themselves of a 4-year-old girl with Dias-Logan Syndrome, they saw the need for a dedicated organization and stepped up to create the very first support network specifically for BCL11A IDD. They're the driving force behind the community we're building today, and are here to share more about their organization, their mission, and future initiatives. Virginie and Alexis, if you'd please give us an update on your organization.
Bonsoir à tous. Donc, nous, c'est Alexis et Virginie. On est les parents d'une petite fille atteinte du syndrome Dias-Logan également, Kélia. Elle a 3 ans aujourd'hui. Et tout d'abord, merci. Je voulais vraiment remercier Amy pour tout ce qu'elle a fait pour cette organisation. Merci au docteur Dias d'être présente parmi nous et pour ces explications qui vraiment ça nous a plu, c'était génial. On a appris beaucoup de choses. Donc, merci à vous pour votre intervention. Je te laisse prendre la parole.
Alors, nous sommes désolés, comme nous sommes un peu novices, nous allons lire les notes et les textes que nous avons préparés. Ça permettra une meilleure compréhension pour tout le monde et que ça puisse permettre que ce soit fluide et bien traduit. Donc, si vous voulez, tout d'abord, nous avons créé un site internet que nous avons réussi à développer. Nous en sommes très heureux de vous annoncer qu'il est maintenant en ligne, traduit dans plusieurs langues. Il a été pensé pour être un point central pour la communauté, un espace pour les familles, les professionnels de santé et les chercheurs. Voilà, c'est le tout début, mais c'est une première étape très importante pour structurer quelque chose à l'international. Nous tenons quand même à préciser que nous sommes parents comme vous, que nous sommes amateurs en la matière. Nous faisons de notre mieux, mais tout est perfectible. Donc, si des personnes ont des suggestions d'amélioration ou quelque chose, surtout n'hésitez pas à nous le dire. Nous prenons les informations avec bienveillance et avec intérêt. Donc, si vous voulez, aujourd'hui, la communauté grandit de jour en jour avec des familles de plusieurs pays. C'est pour cela qu'au tout début, on a peut-être un petit peu insistant avec beaucoup de noms pour la mise en place de la carte de recensement internationale, où à ce jour, nous avons réussi à obtenir la localisation de 109 familles à travers le monde. Donc, 109 familles qui ont un enfant bien souvent ou la personne qui est atteinte du syndrome Dias-Logan. À la suite de cela, le généticien de notre fille connaissait des chercheurs donc, du CHU de Poitiers en France qui nous ont contactés parce qu'ils ont trouvé l'idée très intéressante et ils nous ont sollicités. Et nous, on leur a demandé comment est-ce qu'on pourrait faire évoluer cette chose-là en quelque chose de conforme avec les RGPD, donc les droits de données d'accès et tout ce que comme Cristina Dias l'a expliqué. Et donc, ils nous ont aidés à monter le prochain formulaire qu'on appelle, nous, le Framaform, qui est sur une base de données, donc du coup, qui est anonymisée. Et on a pu récolter 69 réponses de familles sur notre formulaire, dont nous avons un petit peu partagé quelques données sur le site Internet. Il y a pas mal d'autres données. On aimerait qu'elles soient exploitables pour éventuellement une future recherche ou autre. Donc ça sera plutôt à voir avec des chercheurs comme pourquoi pas le docteur Dias ou autre. Il faudra qu'on en reparle et qu'on revoie si ça peut être utilisable ou pas, ce que l'on espère. Si vous voulez, avec tout cela est encore en construction, mais les bases sont là. Nous sommes encore aussi en train également de structurer l'association avec des membres actifs, ayant chacun des rôles et des missions précises. Donc, nous lançons un appel à ce jour. Si des personnes et des familles, ou même, comme l'a dit le docteur Dias, des patients, des enfants, des personnes atteintes du syndrome, veulent s'impliquer et veulent s'investir au sein de l'association, nous sommes tout à fait ouverts et disposés à donner des postes à responsabilité et des fonctions en fonction évidemment des disponibilités de chacun et des compétences de chacun. Mais le but, c'est de vraiment créer une vraie organisation internationale dans l'intérêt de la recherche Dias-Logan et des familles. Voilà, grosso modo, je pense que j'ai un petit peu résumé la chose et j'espère que la traduction se fera bien. Donc, pareil, si vous avez des questions ou autres sur cette organisation, n'hésitez pas à nous contacter. Comme on vous l'a dit, on est novices, on débute, on a mis un peu de temps, on a essayé de structurer juste avec ma femme et moi-même d'abord d'un point de vue légal en France et réglementaire. Maintenant que c'est fait, on peut intégrer toutes les familles du monde dans l'association sans aucun souci. Concernant donc les chercheurs, on va vous dire un petit peu sur quoi on a travaillé. Donc évidemment, on a déjà contacté aussi en France pas mal d'organismes qui pour le moment se cantonnent juste à la France, que certainement le docteur Dias et d'autres docteurs connaissent comme Orphanet ou l'OMIM ou la Fondation Maladies Rares, qui sont des organismes qui aident justement des associations de maladies rares à se développer à l'international et dans le syndrome de Dias-Logan BCL11A-IDD. C'est d'autant plus rare que vraiment tout est étendu sur le territoire international. Donc, du coup, concernant les chercheurs, nous sommes déjà en lien avec plusieurs cliniciens, notamment ceux qu'on vous a parlé du CHU de Poitiers. Les collaborations se construisent progressivement. Notre objectif à terme, c'est de structurer un cadre vraiment formel et notamment, comme une question qui nous a été posée, de constituer un comité scientifique consultatif de sorte que plusieurs cliniciens, médecins, chercheurs pour qu'ils œuvrent tous dans la même direction et qu'ils arrivent tous à s'entendre. Je pense que le docteur Dias me reprendra puisque c'est pareil, on n'est pas non plus encore expert en la matière. On le deviendra un jour avec votre aide à tous, on espère. Mais voilà, c'est le but, c'est que tous les chercheurs et tous les cliniciens aillent dans la même direction et puissent collaborer ensemble et avec les familles. Voilà. Comme l'a dit le docteur Dias, c'est très important, le début d'un registre patient qui serait vraiment le début de toute chose pour le syndrome. Et ça serait vraiment formidable, et on espère vraiment que ça puisse aboutir avec l'aide de tout le monde. Donc sinon, à l'international, j'espère que je ne parle pas trop vite, à l'international, donc comme on l'a dit, la dynamique commence à se développer, et notamment aux États-Unis, où avec EMI, ici présente, la dynamique est en train de se mettre en place puisqu'elle a l'idée de structurer un groupe local pour le territoire américain qui serait en lien avec l'organisation du coup internationale Together Dias-Logan pour avancer ensemble. Voilà. On tient à le redire, l'association Together Dias-Logan, certes on est français, mais le but c'est vraiment de la rendre internationale. C'est pour ça qu'on fait appel à vous de vous investir et de se mettre avec nous. Voilà, elle est juste française parce qu'elle a été créée en France, c'est tout. Mais nous, on veut vraiment que ça soit international. Voilà. Ensuite, on a un autre gros projet, qui est le gros projet de l'association, je pense, pour l'instant, en termes de financement et de financement, on va juste dire ce mot-là. C'est nous allons organiser une rencontre internationale, du coup, certainement peut-être la première grosse rencontre, en France au mois de juin 2027, qui serait les 3, 4 et 5 juin. Donc l'idée, c'est 3 jours. Pourquoi? Pour que les familles, notamment beaucoup, viendraient de loin, puissent s'organiser plus facilement pour venir au niveau transport. Pour que les chercheurs aussi, la journée du 4 juin, donc du vendredi, serait consacrée à une rencontre entre chercheurs, cliniciens et familles, comme un peu l'a fait le docteur Dias, l'explication de ce que c'est d'abord une maladie génétique, comment ça s'applique à BCL11A, parler un petit peu de tous les variants et les différentes choses, parler des projets en cours, de la recherche fondamentale, voilà. Il y a plein de sujets qu'on pourrait aborder, mais ça reste encore une planification à faire. Mais surtout, voilà, on voulait vous parler de ça, que si jamais vous êtes intéressés, on organise ça. L'objectif pour l'association, et c'est nous qui allons travailler avec l'aide de vous, on espère, qui allez nous rejoindre, c'est de récolter des fonds d'entreprise afin de financer ce projet, de sorte à ce que les familles et les chercheurs qui viennent n'aient rien à débourser pour l'hébergement et la restauration. Voilà, il y aurait juste, entre guillemets, le transport, mais l'association essaiera d'aider aussi au mieux pour organiser tout cela. Voilà, donc tout serait sur place dans un village vacances. En fait, il y aurait l'hébergement, la restauration, toutes les infrastructures également et les équipements. Et nous avons également prévu des professionnels pour encadrer nos enfants le jour de la rencontre, par exemple, avec des activités diverses. Voilà pour ça. Du coup, il y a un petit peu de notre partie, et nous avons bientôt terminé. Des données de la recherche, donc le travail important sur le recensement international, comme je vous l'expliquais, la carte des familles. Nous allons continuer un petit peu à le faire puisque ça nous permet de créer du lien justement avec des personnes qui sont un peu partout autour du monde, de prendre connaissance avec eux et de voir quelles sont leurs difficultés territoriales ou dans leur pays. Parce que comme l'a dit le docteur Dias, on s'est très vite rendu compte que déjà, selon le pays dans lequel on est et selon encore même dans le même pays, la zone géographique où nous sommes, nous n'avons pas la même prise en charge, la même chance. Voilà, on va réduire à ça comme ça, la même chance entre guillemets. Donc, l'idée, ce serait vraiment de travailler sur cela pour voir qu'est-ce qui pourrait améliorer, faire un peu comme une fiche type, grosso modo, qui pourrait être mis en place tout de suite dès le début du diagnostic pour que les familles sachent, voilà, il faudrait mettre ça en place, ça en place, ça en place, parce que la majorité du temps, les médecins, même avec nous, et même nous en tant que parents, on n'a aucune information, on ne sait pas, et les médecins que l'on voit généralistes ou traitants ne connaissent pas le syndrome et ou au mieux se renseignent un petit peu, mais sans plus. Voilà. Là, on vous donne notre ressenti à nous et de ce qu'on a pu voir au fur et à mesure avec les échanges des familles. Voilà. Donc, le formulaire que nous avons fait, nous espérons vraiment, comme je l'ai dit précédemment, l'utiliser pour un début de registre patient et que le docteur Dias puisse vraiment affiner tout cela et créer vraiment une dynamique dans ce sens, comme elle l'a expliqué. Donc, on a vraiment très bon espoir vis-à-vis de ça. On est très heureux et très contents. Et pour conclure, du coup, notre volonté est très simple. On veut avancer ensemble avec les familles et les professionnels et construire une communauté très forte pour améliorer la compréhension et la reconnaissance du syndrome de Dias-Logan. On vous remercie beaucoup et force à tout le monde.
Thank you so much, both of you. We've actually had no questions. I just have a comment thanking you both for the incredible work that you've done towards this organization. Like Virginia mentioned, if you have any interest in helping, joining their efforts, please contact them via email or their Facebook. And if you're interested in, if you're in the US, we are starting, like they said, to start a chapter, if you will, in the US. We will share mission. But if you're interested in that, please reach out to me. We're in the very early infantile stages of setting that up. Before we get to questions, I wanted to mention one other exciting event we're trying to put together. I'm currently just in the early stages of planning an in-person family meetup in Orlando, Florida, February 2027. This will be very informal. This location was chosen from a family in the UK wanting to find a spot in the US to visit. So we landed on Orlando, Florida. The date is to be determined, but we're looking at February 15th that week. And I'm hoping this just gives another opportunity for families to meet if they're unable to attend the conference in France in June. I really hope most of you can. The plan is to meet in an indoor play center that specializes in sensory needs. And my goal is to rent the entire facility and admission be completely free for families. To keep the Dias-Logan family Facebook group focused on family support, I created a separate group just to discuss those travel logistics. So if you're interested in that, find that group and join us. Even if you want to just follow along on our progress, all of the updates for that will be in that space. Really, I have no questions that were put in the chat. I think Dr. Diaz did a wonderful job giving an overview that really answered the questions that were submitted early. And again, Virginie and Alexis, thank you so much. I did have something come in, just another thank you. I really appreciate all of your support. If there's nothing else, we're a little bit early on time. Was there anything else, Dr. Diaz or Virginie and Alexis, that you'd like to add?
I was just going to follow up on something that Alexis said, and this is, I guess, more rare disease focused than BCL11A developmental disorder specific, and I do quite a lot of advocacy work in rare disease in the UK. It's important to note that most doctors will never have heard of Dias-Logan syndrome, and that's okay. There are over 8,000 rare diseases. 1 in 17 people have a rare disease, so collectively they're really, really common, actually. In the UK, 3 million people have a rare disease, and yet it hasn't been a priority, but it's starting to be. So we all have people in our lives and that we know that have rare diseases, but we can't expect doctors to know all of the 8,000 rare diseases that are out there. So you will become experts in your child's condition, and you will be the person who's explaining to the doctor what the condition is in many situations. In some cases, if it's a geneticist, they may know and they may have that information, which is great. But GPs, pediatricians, other healthcare practitioners, they're not going to know, and don't hold that against them, because really it's really hard to know all of these rare diseases. They have to know a whole bunch of other things, whereas you're focused on one, which is your child, and which is great. And that's why you become an expert, right? Because that's the condition you're focused on. So it's just having that patience and that empathy with them. And if you do, I think that you'll see that they usually are receptive, and they will want to learn, and they will want to help. Most people, I can't vouch for everyone, but I think most people will.
Wonderful, thank you. We did have one question that popped up. I think that might have already been covered, Dr. Diaz, slightly, but it was a question about splice site variants in particular.
So it depends on the splice variant, right? So that's why I really can't, without knowing, I can't do individual counseling in an online session. It really does depend on the splice variant, because splice variants have very different effects, depending on what the action that it has, and where it has, and what it's causing. So it can be something that is a loss of function, but sometimes it's just a slightly different function. So it really does depend on the splice variant.
Thank you so much. Well, if there isn't anything else, I just want to again point out that this is the beginning of many future discussions and events as we continue to learn and grow together as a community. And I want to thank everyone for attending, and I truly hope today's session was informative. A very big thank you to Dr. Diaz, Virginie, and Alexis again for taking the time out of your busy schedules to speak with us. I will be posting the recording of the meeting next week to the Dias-Logan Facebook group. If you have any other questions or concerns for me, you can reach out to me via Facebook or email. And if there's nothing else, I hope everyone just has a wonderful Friday and weekend.